chapter 11. This is all about growth and change over time. So we're going to stress that with development, we typically think of it in that idea of improvement or growth only. So changing to be more complicated or to do more things. However, we're going to find that development has a bunch of different patterns. So yeah, we do see a lot of advances, especially early on in life, but we'll also start seeing things like gradual declines as we get into later life. And we can tie that into our discussion about intelligence as well. But we're also going to see some weird patterns in the middle, where we're going to see these weird U-shaped distributions. And what that means is when we graph your sort of performance on a task across time, we'll find that individuals tend to do really well, and then they tend to do really poorly, and then they do really well again. And we'll see this for a couple of things in infancy, one of which is our ability to localize auditory cues. And this is really strange, where we'll hear a sound and know to turn towards it very shortly after birth, but around 4 months of age, we lose that ability. We stop localizing, and then it comes back a few weeks later. So we have this strange period in the middle where maybe because our brains are developing and changing and making new connections, we sort of temporarily lost that ability. We'll see the same exact pattern in some of our reflexes as well. One of our stepping up reflexes, again, disappears when you're a few months old and comes back when you are ready to start walking. So we'll see a couple of those odd examples of sort of gaining and then losing and regaining abilities as well. But all of that to tell us that development isn't necessarily just going to be gaining. It isn't always just growth. It's change in a couple of different ways. So we can have things that are progressive, so the growth side of things, learning to do things, being able to do things in more complicated ways, but we then also have those regressive changes, so declines with age. So we'll see not just cognitive declines, but also some of our motor declines as well, and that'll come much later on in the chapter. For our plan for this chapter, we start early on with a little bit of background information, getting ourselves situated with a couple of terms, and also some of the basic research approaches that we often see with developmental studies. And then we'll get into sort of prenatal, natal, and then adolescent, and then old age, different categories, and each category gets its own little section. So we'll have quite a bunch of topics that we'll be touching on, and as per usual, it's a lot of information, but we can't go into as much detail as we might want. So yeah, we'll possibly get to some of those later on. But our starting point, as I said, some of our research designs that are really important for developmental, we have 3 here that we can talk about. And so you'll find that some of the aspects of actually conducting the research within these designs is very similar to what we've talked about in chapter 2. It's actually one of the reasons why I really like redoing chapter 2. But this is saying that we're specifically using this to look at groups as they change across time periods. And there's a couple of ways we can approach that, and then the methods themselves might be experimental or observational, correlational, depending on our setup. But for the big picture stuff, we're going to look at cross-sectional, longitudinal, and sequential research designs. And so each of these is going to get their own slide. And as per usual, I have a couple of different ways to present the information. One term I want to get into here is the idea of a cohort. And this is a group of individuals who all share some kind of trait. For our interest here, these individuals would all be the same age. Usually you're probably also getting them from the same place because you want to try and control as much as you can, but it'll depend on which research topic you have. But for the most part, when we talk cohorts, we're talking about groups of individuals that we might be able to follow across time. So for our cross-sectional research, this is when we're going to measure a whole bunch of people of different ages all at the same time. So this is sort of our, let's get the data as fast as possible. So what we would do is we pick a year and we're going to sample, say, 10-year-olds, 20-year-olds, and 30-year-olds. And that way we get a cross-section of people of those different ages. So we could say that each of those groups would be one of our cohorts. So our 10-year-old cohort and our 20-year-old cohort. And then we can compare between them. And the hope would be that whatever it is that we've measured for our 10-year-olds and 30-year-olds, or 20 and 30-year-olds, when we compare across those groups, the differences we see are hopefully due to their differences in age. We, of course, would have some concerns, hence the whole issues section. And the biggest of these is the fact that these individuals all grew up during different time periods. So if we think about it, 10-year-olds would have been born in 2013, and 20-year-olds in 2003, and so on and so forth. So if you think about how society and the world have changed over those time periods, there might very well be something that occurred during their formative years that's really dramatically influenced their behavior, whatever it is that we're interested in. So when we conduct studies like this, we have to keep in mind that they might have had very different experiences. We might have seen pretty dramatic shifts in culture and society, even environmental changes, depending on how long of a time scale it is that you're looking at. But even, I use the example of myself and my sister. We're only 6 years apart, but she doesn't remember ever using dial-up. She doesn't remember not having a computer in the house, whereas I was old enough to know those things. So very different experiences that might shape how we view technology, which could therefore then be tied into whatever it is we're studying. And if you go back further, think about, say, comparing your generation to your parents' generation to your grandparents' generation. Are there differences that might not just be due to age, but the time periods they grew up in? For sure. So those are things we need to keep in mind. And then for visuals, I tried to do these where different colors are going to be different age groups. And then we're going to see that we have different times, depending on which of our designs we're using. For the cross-sectional, we're saying, okay, at time 1, we measure all of these groups, all of these cohorts, all at once. And I suppose if we're talking about benefits here, because we focused a lot on the negatives, like I said when I introduced this is a quick way to get a lot of data. So you are only limited by how long it takes you to, say, administer a survey or carry out your experiment on all those groups at once. Some of the other techniques, we basically have to wait for time to pass to see how people change as they grow up. So here we get to skip that waiting for time to pass, but we have all of those other considerations to keep in mind. So next we get our longitudinal research design. And so this one is saying, okay, instead of a whole bunch of different cohorts all at once, let's do one cohort across a bunch of times, kind of flipping it on its head. So we're going to test that same cohort at a bunch of different time periods. So we're going to survey them as 10-year-olds in 2000, and then as 20-year-olds in 2010, and 30-year-olds in 2020. And obviously looking at this, we're saying, okay, so now we have to wait 20 years to see how these people are changing across time. So this is like our repeated measures that we talked about before, where we have the same group, we're just measuring them multiple times. So we have the same benefits of our repeated measures, where we get to have less of those individual differences because you're just comparing them back against themselves. The other things that we want to consider, though, the downsides being this takes a really long time. And as we have long periods of time where you have to have people continue to show up, you end up running into things where people drop out. So you'll have people stop showing up, you'll have people who move away, they're no longer interested. If you follow into older ages, maybe people are dying. So you never know who's going to come back. And then, of course, from the statistical point of view, you have to make a decision about what do you do with incomplete data? What do you do when you only have, say, 3 sets of measurements and you should have 4? How can you fill in that gap? And in case you're curious, you don't need it for the class itself, but there's a couple of ways. The most conservative one is to just throw out incomplete data. So we'd say we have to drop this. We do then have that potential of, is there a pattern in people who are more likely to drop out of one of these studies? Because then you can bias your sample by losing certain people. But you also could do something like, we can use other individuals to make an educated guess about what their scores would continue to be. So maybe we use the complete data to make some equations and then make our best guess based on what we've seen in the past from the one with incomplete data. There's a couple of different approaches. Sometimes it's as simple as you just take the average for the group at that time period and sub it in. But different areas will have different standards, and they have to explain why they're using whatever their standard is, because you want to be transparent. But yeah, so time-consuming, people drop out. And then we run into our last thing, which is saying, okay, well, before we were concerned that all of our different cohorts measured at the same time had different life experiences. Well, now these people are all starting from the same time point, but can we actually generalize what we've found with them to other groups? Or is this something that only applies to that particular generation, people born in only that year? And we can't be certain. So if we have these problems, and you might find that some of them are sort of complementary problems, then our solution for that should make sense. But just pop in to see the visuals. This is showing all the same group, but now across multiple times, whereas the first one was the same time, but different groups. Hopefully the colors help. And then our big, let's combine the two and take the best of both worlds, is using sequential research. And so here we have a combination of cross-sectional and longitudinal designs, because now we're going to have multiple cohorts, but we're also going to follow them across multiple periods in time. So instead of just one cohort and multiple time periods, or multiple cohorts and one time period, now it's multiple of everything. And because we're doing this, now we're not limited to, is it just these specific individuals? Now we're not concerned about can we generalize, because we're looking at people born at a bunch of different times. But obviously from this is a lot more intensive in terms of data collection, and we still have that long time period that we would get from our longitudinal studies. So we still have some of those concerns. So it's a lot more comprehensive. It allows us to get around a lot of those concerns from the two before, but you invest a lot more time and effort. And if you look at some of the sort of most famous, most held up developmental research studies, some of them have taken decades to work through. So if we look at some of them, they've followed people for 60, 70, 80 years. So they have long-term data, but you're then saying, okay, well, they had to wait that long. So you can have studies where you can do your, once a decade, you can check in on their progress to see if anything's changed. So it takes a lot of commitment. And obviously they have a lot of dropout over those periods of time. All right, and so here, if you wanted sort of the written way of it, you'd say in 1 year, we're gonna survey people in these ages, and then the next year, a decade later, some of these are the same, so the former 10-year-olds are now 20-year-olds, but now we have new 10-year-olds. And then again, next year, 10 years later, new 10-year-olds, the previous year's 10-year-olds are now 20, and the previous 20-year-olds are now 30, so on and so forth. And so for this one, I found this diagram isn't necessarily as helpful, but I did do a visual this way in case this works better, where our cross-sectional and our longitudinal are pretty easy to see. Again, colors to help. But for the sequential. This is showing that staggering of having those multiple cohorts. You start at the beginning and then you follow multiple cohorts over those time periods. And just for ease, I've been using sort of rounded numbers with 10-year intervals, but you can absolutely do these as check-in every 6 months. Especially in very young children, you can sometimes have these where they want you to come in every month. So it all depends on what it is you're interested in, what your demographic is, things like that. We might also want to have sort of that concern of when we have studies like this with such potentially long time commitments, it can also be really hard to recruit people into your research. So if you wander through the psychology department, you probably see a lot of advertisements for come participate, or especially do you have children, because we have developmental labs and we'd love to have you participate in research. So it's sometimes very difficult to do that recruiting. And again, thinking back to chapter 2, if we looked at recruiting only through, say, posters in the psychology department, what kind of people might we be attracting to participate in our studies? And are they necessarily representative of all people or even all university students? You'd have to see what it is you're studying. But for the most part, we'd say there's definitely a couple of levels of bias there. All right, so those are our starting points, a couple of the different ways that we can choose to sample in order to make these comparisons to look at how stuff changes over time. Like I said, different studies are going to have different preferences, different concerns. Some of them might tolerate some of our downsides of something like a cross-sectional study just to have a quick result. It all depends. And a lot of this chapter is going to be a lot of that. Here's the broad picture stuff, here's what we tend to see, but each study will be different. For some of our developmental paradigms, we're going to look at some of our upcoming designs that we see, and a lot of them end up sharing some pretty similar logic. I guess to take a step back, when we're looking at developmental questions, we run into that issue where for a good chunk of our development, those early years, we're not capable of communicating the same way that we are as adults. So if you have a bunch of adults and you want to see how they change from 20 to 30, you just do a bunch of surveys or ask them a bunch of questions via interviews, or have them come in and tell you what they're experiencing. But if you have a bunch of 6-month-olds, how are you going to determine what they are and are not capable of? So this is where we have these special paradigms, these special designs, to try and get at these abstract concepts. And I find this section really interesting because, as I've said before, people are not my forte for research. But non-human animal research has the same problem, where we can't ask an animal what they are experiencing or what they are seeing. So we typically have to design something where they will react one way if one thing has occurred, and they will react a different way if something else has occurred. And that logic is what we're relying on here, where we're going to set up a bunch of designs that say if they can tell the difference, or if they pay attention to this thing, then we're going to see this outcome, this particular behavior. So we're trying to find a way to quantify these abstract concepts in the absence of the usual questions we might think of. So yeah, what are some of these? And I will say a lot of these working with infants are really odd. But the first of these is a high amplitude sucking paradigm, which is basically relying on the fact that newborns have a whole bunch of reflexes. And if you put something in their mouths, like a soother, they're going to suck on it reflexively. And so you can look at their rate of sucking, so how often are they doing that, and you can use that as a measure of interest. So if something is holding their attention, if they're really intensely interested in something, they tend to suck faster. Whereas if they're bored, they might slow down because they're looking around or not focused. So you can look at those changes in frequency to judge preference. And this doesn't have to tie to, say, food-related things. This can just be like showing them different patterns and seeing how much that's holding their attention or not. My preference are the habituation paradigms, where you might remember, but habituation is relying on the fact that if we see the same thing over and over again, we tend to get used to it. And specifically, we stop responding to it. So in my 104 class, when we talk about learning, I use the example of a fire alarm, where if the fire alarm goes off, they're testing the alarm in your building, and every 5 minutes for the next 6 hours, your alarm is going to go off for a few seconds. The first few times you probably jump quite a bit, but eventually it's going to stop affecting you. And you can still perceive it, you can still hear it, you're obviously capable of hearing the sound, but your response has changed. And so we can use that where if we habituate you to something, we show you the same thing over and over and over again, you will stop responding. This thing will no longer be pulling your attention. If we then introduce you to something that you register as different from what you've been seeing, now all of a sudden that's interesting. It's going to pull your attention again. And so if we see that you respond to some secondary stimulus, then we would make that conclusion that you see this as different from what we use to habituate you. Whereas if something new comes in and you don't react, then you're treating it the same as you did that initial stimulus, and you don't see them as being different from one another. We'll actually get to look a little bit earlier on where we use that exact same technique to determine how clear an infant's vision is at different ages. So we look at how you respond to photos with differing levels of blurriness. This can also tie into things like novelty versus familiarity, where we use something called looking time, where we see how long you stare at something, where if it's something that's, it gets kind of weird to talk about like the habituation where something that you've seen a lot over and over and over again gets boring. We also then, when we look at familiarity, if we say something like your parents over a stranger, in the case of parents, a familiar face is comforting and therefore draws your attention. But if we're looking at, we show you the same thing over and over again, in that case, the repeated exposure makes it boring. It always frustrated me that, like, why is something that you've seen before exciting in one case and not exciting in the other? But it has something to do with that comfort aspect. And for familiar faces, it's not like you're shown the same face over and over and over again in that study. It's more of a, this is something you've seen over long periods of time, just in case you're going to start asking questions about that, because I know it's thrown other people off as well. But yeah, we have certain things where if it is familiar to us, we find it comforting. And they've used studies like that play into that for things like, do you remember hearing this song in utero? And you'll typically see that they recognize songs and voices, and even things like if they've heard a particular story before. It can be said in a new voice, but sort of the patterns that are involved in that story become familiar and comforting. So yeah, it's all about which is it that we care about, that familiarity or habituation to stop responding. But both are or can be measured using things like looking time. And we'll also then get into our violations of expectation, but those are really weird and will benefit from having a visual. So first off, I want to look at that looking time and habituation setup. So this is one design. As per usual, I have borrowed this from one of my other classes. This is from one of my learning classes. And this is showing visual fixation in infants. So visual fixation is just how long do you stare at this thing, hence looking time. But they show an image, and here they have two different images of differing intensity. And the graph here is actually showing how quickly we habituate to it. So how long do they look at it before they look away, before it becomes boring? And so a higher number on this axis is saying they're looking for longer. And as we have more trials, that's just how often are we showing this image to you. And these would usually be a few seconds apart. And so with our open circles, that's the more complicated pattern. And typically, more complicated visual stimuli are going to be more interesting. That complexity makes it cool to look at. And so we actually see this sort of spike at the beginning, which if you go into learning, you'll learn that that's sort of a brief sensitization spike where it becomes extra interesting because it's complicated. We do not need to know that. But what we care about after that is, yeah, we still see that habituation, but it's a much slower process. Whereas if we have our simple stimulus, the closed circles, we're seeing that that drops off a lot faster. And even once they're used to them, they look at the less complicated stimulus for a lot shorter periods of time. If we had only shown them, say, the simple stimulus, and then we switched it out and showed them the complicated one, if they have a different reaction, which my expectation would be showing them the complicated one when they've never seen it before, we'd see something like this all the way up here. And seeing that would tell us that they can see the difference between a simple stimulus and a complicated stimulus because they reacted very differently to them. You can also do this with all sorts of other types of complicated and less complicated stimuli. We'll even look at a diagram later on that shows their preference for faces. And the fact that we see preference for even abstract faces comes about very, very young, where young children would choose to stare at a simple cartoon face over something that's maybe brightly colored or even patterned. Faces pull our attention from very early on. All right, so the next bit here, this is getting into those violations of expectation. And there's a bunch of different ways to test these. But for this one, what we're doing here is we're still habituating them. We're showing them that this is something that happens and this is how it happens over and over and over again. And it's kind of hard to figure out what's going on just from the images here. But what they basically have is a flat piece of wood. And there's a hinge on one side. So the hinge is towards the middle here. And what they do is they lift the piece of wood, follows its trajectory, and it falls to the other side and lays flat on the table. And so if they're habituating the child to this, they're going to do it over and over and over again. And we can still look at how long are they staring at this. So are they paying lots of attention? Are they not paying lots of attention? And you can also do this instead of just like the looking component. You could also just say when they turn their heads. But we have that judgment of how long is it holding their attention. Once we're sure they've habituated, then what we do is we place a box behind where that board is going to fall. And the idea being that if the child understands that that is a solid box, that our board shouldn't be able to fall through it. The board should push back and hit the box and stop. And so the other important part here being is that once we've lifted it up above eye level, the child now can't see the box. And this should immediately get you thinking about object permanence, where there's this idea that up to a certain age, as soon as something vanishes from sight, we forget that it exists. So with a lot of babies, if you put a blanket over their toy, all of a sudden they lose interest because they forget that the toy exists. And so if they don't have object permanence, then they would anticipate that as soon as they forget the box exists, it's going to do the same thing it's been doing so far. It's going to fall flat. Whereas if they understand object permanence, even though that's been blocked from view, they would anticipate that the board is going to fall back onto the box and stop early. And then we can have sort of one of our trials as a trick, where as soon as the board is lifted enough to hide their view, you can remove the box. And in that case, when it falls flat, If that's surprising to them, if they weren't expecting it to behave like that, because they anticipated the block stopping the movement, then we'd say that they do have object permanence. They knew that the block was there, and this impossible event was surprising to them. And how do we measure surprise? They stare at it for longer. It's going to hold their attention more. So when something is unexpected, it's exciting and it holds their gaze. So in this situation, if they react differently, then they have that concept of there should be a box here and it should stop the motion. But if they don't have object permanence, then this impossible event wouldn't have surprised them and they wouldn't react any differently from any of those habituation trials. And so you can compare their reaction to different types of these events and make that determination of what they do and don't know. There are other variations of this, and once again, they've done this in non-human animals. I've seen a similar one with both dogs and gorillas, of all things. But what they do is they'll have a screen that blocks your view, and they'll have a couple of ramps. The idea being that you would roll a ball down the ramp so it disappears in the top corner and reappears in the bottom corner. And if you understand that the ball continues to exist, you would then anticipate it exiting in that bottom corner. And you can show them over and over and over again that this happens, and then maybe there's a trial where the ball comes out somewhere else or doesn't come out at all. Are they now surprised? Do they spend a lot of time looking at it to figure out what goes on? And sometimes, especially when you work with some of the other primates, they'll actually go and investigate. They're so curious about what the heck happened that they want to go and see what's gone wrong. And in those cases, once again, if they treat that as surprising, as different, that shows you that they had that concept, that anticipation present. Another recent example of this on social media was when people were doing that trick with the blankets for their pets, and they would hold it up and then disappear behind it and drop it. And you'd find that different species would be more or less reactive. That can also be a measure of object permanence. And you'll find a lot of variation, but I especially enjoyed, as per usual, all the bird videos, because a lot of parrots get very vocal when their people vanish. But the dogs were pretty neat too, because some of them do sort of expect them to be there. All right, so those are a couple of things that we can look at. Some other ones are the false belief paradigms. These have a whole bunch of different names, but there's a bunch of conventions that you typically see over and over again. False belief paradigms are used to measure theory of mind. And we'll talk more about theory of mind later on, but as a brief introduction, theory of mind is just this idea that you understand that your own thoughts and your own knowledge and your own internal processes are yours, and that other people can have their own thoughts and their own knowledge that can be separate from yours. I usually point out that with children, there's usually an age where they start being able to lie. It's like they've figured out that this is something they can get away with, and that's when they develop their theory of mind. Before that, they don't try to lie because they assume that if they know they're not telling the truth, you know they're not telling the truth either. Whereas once they've developed that theory of mind, now they're going to sort of understand that I can withhold information, and if you don't have the true information, then you don't know that I'm lying. And so there's that age where when they figure it out, it's new and shiny and exciting, so they tend to do it very often. But some of our assessments for this are seeing, do you grasp that idea that you might have information that someone else doesn't? And I don't think I have a slide for elicited imitation paradigm, so I'm just going to do that here before I show you an example of the false belief paradigms. But elicited imitation is just what it sounds like. It's you get somebody to copy your behavior when you've told them to copy that behavior. And it's specifically used to see if they can remember the actions that you carried out. and could then do those themselves. So it can get into things like muscle movement and can you copy those specific behaviors, but it can also be tests of how long you can remember things, especially if they maybe distract you in between. But we won't talk too much more about those. The other ones are used a whole lot more, at least for our discussions. All right, so as promised, one of our false belief paradigms, they call this the Smarties task, which has always bothered me, just a little bit of a rant. So in Canada, Smarties being the chocolates that are coated in candy. In the US, Smarties are what we call rockets, neither of which is what this box looks like. So I always include it because I either have very useful diagrams or ones that make me mad for some reason. But for this one, we have the researcher and the child, and you show them this box and it says Smarties on it. So something the child is familiar with. And you say, okay, what is in this box? And they're going to assume that the outside of the box is truthful. They don't have any other reason to think you might have something weird in the box, so they say Smarties. And then we get them to open the box. They see that it's not actually Smarties, it's pencils. And then we're saying, okay, you've learned new information. It looks like it's Smarties, but it's actually pencils. So let's close the box back up and let's go show this to somebody else. If we go show this to your friend Jenny, what's she going to say is in the box? And if the child has passed that theory of mind threshold, they would say, well, she's going to assume there's Smarties in this box because that's what I assumed, and there's no reason not to think there's Smarties here. Whereas if you don't have theory of mind, then you're going to say pencils because you know there's pencils, and therefore everyone knows it's pencils too. Another variation of this that you might have seen is involving like a basket and a box and two kids with a ball, where one of them, it's usually Mary and Sue, and so Mary will come in and she'll put her ball in the basket, and then she leaves, and then Sue comes in and moves the ball to the box, and then she leaves, and then you ask the kid when Mary comes back, where is she going to look? And she should look in the place she last saw the ball, but if children can't pass this task, they'll tell her to go look in the box because they know that that's where it is. One of the really cool parts about that, though, is it's presented as sort of a pass-fail, where you can either figure it out or not. But more recent research, they've started looking at things like how people move when they're answering these questions, and especially eye tracking. So for that, is it the box or is it the basket? Children will look to the correct location. So they look to where they should say it is based on that person's knowledge, but they still say the wrong location. So there's this window where they're almost having that grasp of theory of mind, but it's almost like they can't inhibit saying the wrong thing. So they need to tell you where it actually is. They can't hold that back to tell you where that person thinks it is. And there's a bunch of research that gets into this and why that might be the case. There's been a couple of theories that actually link this to individuals with autism spectrum disorder, where it was originally thought that people with the disorder would actually fail this task more often because they have trouble putting themselves in other shoes. But depending on how the task is framed, sometimes they end up performing better. It really just depends on which paper, which research study it is that you're looking at. But yeah, the extra stuff in the background isn't super important, more just here's how they work. But for those who are interested, I always include little tidbits. All right. And this slide here, this is just sort of a reminder that we've mentioned before, questionnaires and surveys, those are pretty common methods for psychology. They are massively employed within developmental research. So the stuff we were looking at before was a whole bunch of different behavioral measures that can give us those hints of what's going on internally. But once we've passed into older ages where you can ask questions, they like to do that as well. So you'll sometimes see a combination of all these different methods, But especially when we're trying to collect information about what people see or what they think or how they feel, having those explicit reports can be really useful. Sometimes what we'll do, is, even with children, is instead of having the children fill out a survey or a questionnaire, you can actually design it to be filled out by the parents or the teachers or guardians or whatever, someone who interacts with the child regularly. But we've always mentioned that any kind of self-reported data, we have to take it with a grain of salt because people tend to be biased. And parents tend to be very biased about their own children. So they might overestimate their abilities because they're so proud and they must be advanced. Or we run into issues of maybe they don't remember accurately how often something occurs. We have that availability bias where if something is sort of noticeable or memorable, we might assume that it happens more often than it really does. So there's a couple of things that can bias our research from that point. So yeah, our memories aren't great. Biases are definitely possible. Sometimes we'll use like remote sampling methods. So maybe you have cameras set up somewhere and you can check in to see how often certain behaviors are occurring. But for those types of studies, there's the whole issue of privacy. So there's lots of considerations in the background. Which ties in nicely to our ethical considerations. So same as our end of chapter 2 stuff, we have to get approval from those institutional or internal review boards, especially when you're working with these groups that wouldn't be able to advocate for themselves. So when working with children, they are very strict about what you can and can't do. And for those studies, the parents need to be thoroughly educated, so you get that informed consent, as we said before. One of the sort of best practices, not necessarily required, is to try and get assent from the children if you can. Basically, get them to agree to do the activity with you. And in a lot of this research, it's something like, will you come play with the blocks with me, or something like that. And it's mainly because it's really hard to work with an uncooperative kid, so asking is going to get them involved. But yeah, there's a whole bunch of ethical stuff in the background to consider. We also have to have those debriefings afterwards. As per usual, if anything was withheld, you have to have a debriefing. Mainly, there's always something withheld. So they're probably always having a debriefing. But it's especially important when you're working with children of like, hey, here's what we did, here's why we did it, here's maybe something to watch for or to expect, just to make sure that this hasn't negatively affected them in any way. We also have to have that consideration of what is it exactly that we're testing or measuring, because you can't manipulate certain things. And even if we go into a little more abstractly, if we get into things like how does, from our correlational studies, we were looking at things like how does growing up in a low socioeconomic status household affect whatever? Or how does having lived in an abusive household affect whatever? In those situations, there's actually rules about if researchers should sort of step in and alert authorities and things like that, because you can't observe bad things happening. It's one of those where if you are getting those types of studies, it's after the fact, something that wasn't possible to intervene in. I know that's something I go into a lot more detail in methods courses, but I don't think we touched on it here. But definitely something to keep in mind on that ethical standpoint. Before we get too far off the beaten path, our practical considerations, we already talked about the difficulty of finding participants. It is very hard to recruit people. Like I've said, we use our convenience samples of our research pools, but that means that we have lots and lots of people of university age and very few people of any other demographic. So we definitely have some of those limitations, and a lot of that leads to fairly biased samples. So we have to be pretty cautious in those situations in how we interpret our results. So that leads to those homogeneous samples where they're often people who, say, have an association with the psychology department, or they do a lot of recruiting at, say, hospitals. So people come into the maternity ward to have their children, and there's sort of questionnaires or things available, posters everywhere. And so it would be whoever happens to be in that hospital at that time. So yeah, considerations in terms of bias there. And all of that, having a non-representative or biased sample, can definitely affect our generalizability of those research findings. So not only were we concerned about things like generalizing with our cross-sectional work, we can be concerned about the samples themselves, even if we did one of the better designs. So we have a lot of critical considerations in the background, which by now should be sort of second nature. We've done this enough from all the other topics. We're always thinking critically about what else do we need to be aware of. All right, so our next section here, we're finally getting into prenatal development. So we're going to run through some of the very basics of going from fertilization all the way up to birth. And for this section, I always stress, like, you can take entire developmental biology courses where you go into this in far more detail. However, we get 10 or so slides, so we're just hitting the highlights and the high points and then moving on very quickly. But our overall view here is we're going to talk about our germinal stage, where our organism is called the zygote, up to 2 weeks. So that's going to be from fertilization to implantation. Then we get the embryonic stage, and now it's not a zygote, it's an embryo, so 2 weeks to 2 months. And then we get into the fetal stage, so now not an embryo, but a fetus, 2 months all the way through to birth. And each of these stages is sort of recognized for different qualities, different specializations. And just looking at sort of the changes here, we'll find that there's really rapid development where we go from a single cell to many, many cells to things that have sort of visible features very, very quickly here. And this shows some of that progression where we start with even early expressions, things like smiling and frowning, all the way back at 12 weeks. And then heartbeat getting stronger, things like our visual and auditory senses starting to develop. These are kind of interesting because we'll talk about it, but our auditory senses, those are developed and then being used even while still in utero. So in this situation, this is why you can recognize a mother's voice after birth, because you've gotten used to hearing that voice. Our visual system does also develop, but we don't have exposure to light stimuli. So we don't have that refinement yet. So while we're born with pretty good hearing, our vision is absolutely terrible to begin with. And it'll very rapidly refine and get better, but because of that lack of stimulation early on, we don't have the best functionality there. Into the later months here, or I guess later weeks, is when we start seeing a lot more refinement and fine details. And this is the sort of stuff that will look at the cutoff for, say, survival or viability for premature births. But some of this could be completed outside of the womb. So even if you are born premature, your development will still continue and you'll catch up to other babies around the same age. There's just a little bit of a lag to begin with. And we'll find that for one of our diagrams, we'll look at how things like visual acuity can catch up. So we have a graph where we'll have children born with cataracts. So their eyes don't have the chance to develop until they get them removed. And they pick up to normal levels very, very quickly. So we'll find over and over again that in our early years, in these early stages, our brains are very flexible. So we'll talk a lot about plasticity and reorganization, where our brains and other parts of our bodies can change and grow and adapt very well. But yeah, we'll come back to a bunch of that. So if we're starting at fertilization, probably remember this from bio classes, but we have a sperm and ovum, an egg cell, end up fusing to create a zygote. And if you remember back to talking about chromosomes and some of the bio stuff, we start off receiving sort of one set of chromosomes from our mother and one set from our father, so that when they come together in that fertilized cell, that's when we're going to have two sets of all of our chromosomes. So we end up with 46 total, and they exist in pairs, so 23 pairs. And once that fertilization occurs, it actually occurs up in the fallopian tubes, and then eventually we have implantation. And that's that division between our germinal stage and the embryonic stage is about the 2 weeks that it takes to reach implantation. And during this stage, once fertilization has occurred, our key characteristic is that we have very, very rapid cell division. And so we can talk about it being a blastocyst now, because we go from 1 cell to 2 to 4 to 8 and so on, until we're at the point of having thousands of cells created per second. It ends up being ridiculously fast. But it doesn't form into a solid mass. It's a hollow ball, and that hollow shape is going to end up having these cells migrate in fairly predictable patterns. And once again, if you get into developmental bio, they can look at early locations of these cells and let you know what they're going to become later on. So one of the things that we'll recognize early, if you get to look up close, are things like a neural groove. It ends up being sort of like a dent, a crevice along one side, and that ends up folding in and making your spinal cord. And one of the ends up expanding, and it becomes your brain. But we can see those shapes very early on, and then it just takes them quite a while to develop and refine. All right, like I said, very brief on a lot of this. We can revisit our discussion about our types of twins here. So we've mentioned them before because they were really useful in talking about how closely related individuals are and what that can tell us about the genetic contributions towards some variation in a trait when we were doing our heritability discussions. But twinning actually happens during this germinal stage. So our dizygotic or fraternal twins, they occur when we have 2 eggs that are released during ovulation. Usually only one is released at a time, but sometimes both sides will release at the same time, and that means that there are now 2 eggs that could be fertilized separately. So this is why when we talk about those dizygotic twins, dizygotic because now there are 2 zygotes, but we also talk about them as sort of siblings that happen to be born at the same time, specifically because it's 2 separately fertilized eggs. So they only share 50% of their genes because that's what all of our siblings would share. We have 50% of our genes in common as long as we don't come from the exact same fertilized egg. And that's exactly what happens with our monozygotic or identical twins. And monozygotic because it starts as a single zygote, and then through some fluke during division, they're going to separate out into 2 separate balls of cells. And because they started from the same single cell, they're going to be identical. They're going to have the same genes. If you look into it, so dizygotic, the fraternal twins, there is a genetic component. So you might have heard of twins running in a family, and it could be related to hormone levels that stimulate ovulation. It can also get more common as you get older, simply because, again, fluctuations in hormones. But it's kind of neat to see some of the patterns in when these tend to occur. And different groups can have different rates at which this occurs, possibly because there's maybe some genes, there's versions of some genes that might, again, cause fluctuations. One of the things to note for our dizygotic twins is that they can be different sexes, but don't have to be. So you can still have 2 males or 2 females, but you can also have 1 male, 1 female. Whereas the monozygotic, they always are going to end up the same sex because they have identical chromosomes, and therefore they're going to have the same sex chromosomes. So for females, 2 X's, for males, an X and a Y.